Hi everyone, you are watching Biology with Insha. So in today's video we are going to be talking about the organelles that are found in both an animal and a plant cell. Now this is the part 2 of the video of the AS Biology 9700 of the very first chapter, which is cells and microscopy. Okay, so we have covered only a number of organelles that are found in both an animal cell and a plant cell, and we are continuing with that, and we will be done by the organelles found in both the cells by the end of this video. So in the next video that I'm going to upload a bit about next week, I'm going to be discussing about the organelles that are found in only a plant cell. So I'm going to make it separately to kind of avoid confusion, and then after that I'm going to make a video on microscopy in which we're going to discuss a number of important things, and then after that I'm going to be making some past paper question videos which will be on paper 1 and paper 2. Well, not on paper 3, but I may make some videos on paper 3 as well, including solving some past paper question videos after performing the experiments. Okay. So remember that we are watching AS Biology 9700, right? And I'm also a teacher of O levels 5090 as well as IGCSE. And I've done the past paper questions myself personally, and I have experience in teaching the following subjects. So I hope you guys enjoy this video. So subscribe to my YouTube channel, like, comment down below, and for Facebook viewers, like and follow my page, and of course comment down below, and I'll be taking all the requests I get. Your support means a lot to me. Your comments, your support, your reactions, I get to learn from them, and I really want to make biology easy for you guys. So let me know if I can make it easy in any way. And one other thing I wanted to say is that whatever I teach on my videos, it's all as per the course, as per the syllabus. Nothing is out of the context, so I'm very strict with the syllabus, and I have some amazing keys and past paper golden techniques that I will be telling you as I make videos on past paper question videos. And without further ado, let's get started. Okay, so we're going to talk about the we're continuing in fact with the mitochondria, right? So the mitochondria is found in both an animal cell and a plant cell. Now I'm slowing down now because this is going to get a bit complicated, so I want to be as per your pace. Okay, so the mitochondria has a double membrane, and we know that anything that has a double membrane is referred to as the envelope. Like the nucleus also had a double membrane, as we discussed in the part 1 of the video, so we refer to that as the nuclear envelope. So we can refer to the membrane of the mitochondria as the mitochondrial double membrane or envelope, however you wish. They both mean the same thing. Okay. Now they may ask in the past paper paper 2 that name an organelle that has a double membrane. So you may write mitochondria or chloroplast, or maybe even nucleus, depending on your wish. So you will get 1 mark for that. So just keep in mind, I'm going to tell you this in every step of the way of what is important and what you should remember and what you don't need to remember. Before we move, let's first have a look at the mitochondria in an animal cell. Now in the next video, which I'll make next week, which will be on the organelles found in a plant cell only, we'll be studying a similar structure of a plant cell, of a very complex and a detailed plant cell, in which I will be locating organelles like the rough ER, Golgi body, mitochondria in the plant cell, so that you're able to differentiate between an animal cell and a plant cell, right? I don't have the plant cell right now, but we'll be focusing more on it in the next video, so do not worry about that. And in the video of microscopy, I'll be showing you some micrographs, photomicrographs, and electron micrographs of different parts of the cells. Okay. So I'm grabbing a highlighter here to highlight our mitochondria. Okay. Here we go. This is your mitochondria in the animal cell. Now this here that I'm highlighting in green is your mitochondria. You can see it's a bit sausage or oval shaped. So the key to identify the mitochondria is that it is shaped like a sausage or an oval. It's long, elongated, and I'm just circling it for you guys right here. This is your mitochondria. Okay, so it is a very important organelle, as we're going to discuss now. Okay. So this is actually a bit of a this diagram here is even a more complex and a detailed structure of the mitochondria that you need to know as per the AS level course. Okay, so the mitochondria has an outer membrane and the inner membrane. So when I say the outer membrane and the inner membrane, this here region here is the, oops, sorry, let me just grab a highlighter instead. I don't want to confuse you guys. Yeah, so this here is the outer membrane. It also says your outer membrane here that you can see here, and the inner membrane here. Let's have a look at this a bit more carefully. You see these regions here that I'm highlighting? Follow my cursor. These are your inner membranes, right? So note that it's not directly attached with the outer membrane, it's within. Okay, so basically space between the outer membrane and the inner membrane is called the intermembrane space. So let me just grab a pen this time and show you what I mean by that. What I mean is that you see this space here, you see that red line I'm drawing? This actually is the intermembrane space between the outer membrane, and this here is the inner membrane space between the outer membrane here and the inner membrane here, and this here is your intermembrane space. So folding of the inner membrane is called the cristae. So you see that the inner membrane here is folded. You can see here, follow my finger here, these folds here, right? Like they've been folded, right? The inner membrane is not normally what the students would expect is that they'd expect it to be like this, something like this. Maybe this is the out. Oh, sorry guys, I'm really sorry. Okay, yeah. So they'd expect this to be the outer membrane and the inner membrane to be something like this. It's not as simple as that. It's actually a bit more complicated in the sense that the inner membrane is folded. So this inner membrane here, as you can see, is folded. As you can see these folds here, right? So this folding here is called your cristae. Have a look at this word here, cristae, the foldings of the inner membrane. So I'm going to highlight the word cristae in the diagram here so that you remember what I mean. This here is the cristae, and this is what I mean. These folds here that I'm kind of highlighting right now. This, and this, and note that this is done by the inner membrane, not the outer membrane. The outer membrane is outside the brown region here. The full brown region is the outer membrane, and this maybe I don't know which color is this, pink, I guess. Yeah, right, light pink, something like that, is your inner membrane. Okay. So within the cristae there is a solution, there is a space called the matrix in which the process called respiration occurs. Oh, I'm really sorry, I might have rubbed it out by mistake. Now first I'm going to rub all these not needed content because I hope they have been understood. If not, you can always rewind back, or you can just read through these main points that I have written down very carefully just for you guys. So the matrix is basically the space, right? It is the space between it's actually the space within the cristae, right? It is you can see here, right, this blue region here that I'm coloring with a red highlighter here. It's a solution. There's a liquid in here. So this is here is called the matrix. Even here, this is the matrix. Even till here, this is the matrix. And yeah, matrix, and I'm highlighting it right here. Okay. So basically this is where respiration occurs. But just to remind you, respiration is the process by which glucose is broken down or oxidized by oxygen to produce energy and some other waste products that include carbon dioxide and water. Okay, so the outer wall here of the mitochondria has pores which allows exchange of substances between the cytoplasm and the mitochondria. So just like the nucleus had nuclear pores through which substances could enter and exit the nucleus. Similarly, the pores in the outer membrane of the mitochondria allow substances to enter or exit the mitochondria between the mitochondria and the cytoplasm of the cell, of both an animal cell and a plant cell. Now this here, guys, before I move any further, there's something very important that I just wanted to show you, and this comes in past paper question videos, right? It always comes in past paper question videos, so listen to this carefully, take a note of this, or just watch my video again, whatever is convenient for you. So the mitochondria is very special because it has its own circular DNA and 70 S ribosomes. Ribosomes are basically protein making factory. Let me elaborate. The mitochondria is able, write this down as I say if you think it is convenient for you. The mitochondria is able to change shape and move, right? so it appears differently under the microscope. This came under the past paper question video in 2017. And one other special thing about it is that it has its own circular DNA in the matrix, and it has its own 70S ribosomes in its matrix. 70S is referring to the size of the ribosomes, which is about 20 nanometers, right? And ribosomes are basically organelles that produce or make proteins. We have discussed it in the previous video, and just to give you a bit reminder, let's go back here. And do we have ribosomes here? Yes, these are your ribosomes here. These dot-like structures that produce proteins are also found on the rough ER, as you can see here, not on the smooth ER. That's what makes both the organelles different and gives your rough ER a rather rough appearance. Okay, so just remember this and note that they're all found in the matrix. And yeah, so this here just an equation of cellular respiration. Just to recap, for those who don't know, that's okay. So this is glucose, right? Monosaccharide. Remember, their energy is released in the form of the monosaccharides, never in the form of disaccharides or polysaccharides. This will also come in the second chapter, which we will be discussing. Oxidized by oxygen, which produces ATP, which is your main energy carrying molecule, adenosine triphosphate. It is a molecule that contains energy that breaks down to give you the energy you need. And then there's some waste products like carbon dioxide and water. Okay, this is balanced. You need to know this. This may come in the exam. Keep your mind open when you get something like this. Okay guys, now we're starting off with the next organelle, which is the microtubule. The microtubule is found in both the animal cell and a plant cell again. But this is going to get a bit complex, so bear with me. I'm going to do my very best to make it as simple as I can just for you guys, okay? So the microtubule, before we start, let's locate the microtubule in the animal cell here. Okay, they've shown centrioles. Microtubules and centrioles have a connection here, but I don't think they've shown the microtubule here, so I'm just going to draw it for you and show you what I mean by it. So microtubule is basically a long, hollow cylinder like this. It's not that long, but, well, yeah, I'll show you how long it is. It's about not 500 nanometers, that's the size of your centrioles, something like this. It starts usually like at the edge of the cell and goes all the way to the other edge of the cell, right? Something like this. It's a cylinder, it's hollow, it's long, it's hard, it's usually near the centriole, which is right here, and the nucleus, which is right here. So this is what you should be expecting a microtubule to look like. You can find it in the book of Mary Jones, you can find it on Google. I hope this is enough to give you the rough idea as to how long it should be. From one edge to the other edge of the cell. You can expect there to be about 2 microtubules or so in an animal cell. Okay, yeah. So then again, there's a bit more complicated, complex picture here that you need to know. So it is a long hollow structure present in cytoplasm of both an animal and a plant cell. Now what's the function? The function is to maintain the shape of the cell. Since it starts from one edge of the cell to the ending edge of the cell, it maintains the shape of the animal cell, right? It holds it together as it is responsible for the cell's cytoskeleton, right? This is simple. So let's talk about its composition. It's made from a protein, and that protein is called your tubulin. Your tubulin is a type of protein that is responsible for making your microtubules, and there are 2 types of tubulin that make up your microtubules. First one being alpha, second one being beta tubulin. Alpha and beta tubulin, right? Okay. Let's have a look at the diagram first, guys. So this full thing here that you can see here is your microtubule. It's hollow, long, right? This is the cross section of how you should expect it to look like. Now it's made from 2 particles. Here we have the alpha tubulin. Here we have the beta tubulin. One other extra information I'm giving you here is that your alpha tubulin is negatively charged, and your beta tubulin is positively charged. Since they're oppositely charged, they are attracted to one another, and well, we'll get into a bit more detail as to what I mean. So how are microtubules formed? Let's understand that in detail. So alpha and beta tubulin combine to form a dimer. What do you mean by a dimer? The word dimer basically means when two particles combine together. And in this case, alpha tubulin and beta tubulin combine together to form a dimer. And this here is a dimer that you can see right here. Okay, this here is a oops, sorry. Yeah, this here is called a dimer. And then these dimers join end to end. Let me just explain it here. These dimers join end to end to form a long filament. Now let me explain what is going on here with a pen, of course. Let's suppose that this is one dimer and this is the other dimer. Okay, this is the alpha, this is the beta. Then we have even more alpha and beta, like more dimers. This is one, more joining together like this. So this is one dimer. This is the second dimer. This is the third dimer. So they both join together in a straight line called a what? Filament. And if you want to understand it in a much better sense, just grab a highlighter. This is something of a filament here. This long straight line here is a filament here. See? You can see that in this you could find many dimers here composed of what, the alpha tubulin and the beta tubulin, right? So that's something has been understood. So filaments, dimers combine to form filaments that combine to form your microtubule. So when many of these filaments join alongside each other in a hollow or in a cylindrical form, right, then they form the microtubule. So one filament, many filaments join alongside each other in a circular cylindrical pattern, microtubule is formed. Okay. So what is the function of the microtubule? It plays a role in nuclear division, right? So what the microtubule does is that it makes the spindles needed for the separation of chromosomes during cell division. Now you don't need to get into much detail of this as to what spindles are, because this will be covered in one of the upcoming videos, as we do have mitosis and everything in AS biology, and we will be studying about spindles and everything. So for now, just understand that spindles are something like this, that I'll just explain it here briefly. That these microtubules here will make these long structures, something like these long structures here. These are spindles, and they will kind of separate the chromosomes from the nucleus for cell division. Just explain it briefly. It's not needed right now. It won't come much in detail in the past paper, so don't worry about that. Okay. They form centrioles. So these microtubules that we discussed form centrioles. What is MTOCs? MTOCs is known as the microtubule organizing centers, is actually the location where microtubules are made or destroyed as per the need, right? They can be anywhere in the cell, usually closer to the nucleus. Okay, coming to centrioles. Now centrioles is slightly complex, and you can see there's not much written there because I have to do most of the work explaining it rather than having to write information down. So centrioles are also hollow cylinders that are present in animal cells only. Now these are only present in animal cells. They are absent in plant cells. Remember that, right? I'm just gonna kind of highlight this part here because this is important, that only present in animal cells. So they play a role in nuclear division. Yes, microtubules make centrioles that play a role in nuclear division. So microtubules indirectly play a role in nuclear division by making spindles and blah blah blah, right? So the centrioles lie in a region called the centrosome. Now this is an animal cell. This is the nucleus. Now if you look closely, this dotted region here is the region in which the centrioles are found, and that region is called the centrosome. Okay, centrosome, right? And you can see that in the centrosome you have your centrioles. These hollow cylinders here are your centrioles that you can see here. And if you want to have a magnified look of your centrioles, this is your centrioles, right? And note that your centrioles are made from microtubules, just like we discussed before. Microtubules form your centrioles. So we'll get into how microtubules form centrioles in just a second. Let's first understand this part carefully. But before we understand that part carefully, let's first understand where your centrioles are in a plant cell. I'm going to rub this out. Then again, not needed. rubbing it out for you guys. Okay, here you go. This is your centriole. Here you go, centrioles. Let me just highlight it then. Yeah, oops, sorry, happens all the time. Yeah, centriole is right here, and this is something what the centriole looks like. It's cylindrical as well, made from microtubules. Now this region in which the centrioles is present is your centrosome. This is the centrosome here, right? Just like we discussed a couple of minutes ago. And also note that the centriole, the key to finding the centriole in any past paper question video is that they're always found close to the nucleus. So remember that they're found close to the nucleus, and they're usually found in pairs, two of them together at right angles. You can see that this one here is at right angle. You may sometimes find like one centriole at the top of the nucleus and the other at the bottom of the nucleus. So remember that. Okay. Where were we? Yeah, so what we were discussing was that okay, let's understand this part in a bit more detail. Made from triplet of microtubules each. Complete, okay, I'll get to that in a second, but complete function is still known to be a mystery, although we know that it plays a role in nuclear division. The complete and very precise function is still known to be a mystery. Okay? Okay, so I discussed a couple of minutes ago that your microtubules are basically, your centrioles are basically made from microtubules. How? How is that? So we discussed how microtubules are formed. That's another thing, so go back if you did not understand that. If you still did not understand that, comment below and I'll explain it again, right? Or message me personally via my Facebook page. My team will reach out to you and I will take those requests, okay? Okay, yeah. So, yeah. So it says here, a triplet of microtubules. What this basically means is that, let's have a look at this. This is a cylinder. I'm sorry if this doesn't look good. My drawing isn't that good, but this is a cylinder. And in the cylinder, it basically is somewhat this. And when I say triplet of microtubules, what I mean is that, they're basically, I'm going to use a different color pen, and there's already a blue one at the top. Okay, let's use yellow. No, red, yeah. So this is basically your triplet of microtubules. So there are 3 microtubules. You can see here, there are 9 triplet of microtubules. Sorry, I forgot to write the word 9. So there's 3 microtubules here. Each, this is 1, this is 2, this is 3, making it what? Triplet. This is your first triplet, second triplet, and there are about 9 of these, right? 9 of these, so 3 9s are 27. There are 27 microtubules in total that make up your centriole. Like this is the first one, and the second one, and the third one. But they're found in like 3. They're found in 3 pairs, not in 3 pairs, but like as triplets. Sorry, that's the correct word. They're found as triplets. So that's why we say 9 triplets. So they're like found like these, and there are like 9 of these in a cylindrical form, right? So just have a look at this. No need to know these words. We're in AS level right now. This is not needed. If this is needed, then I'll let you know in one of the upcoming chapters on cell division. Okay, so let's just rub this out. Okay, so you can see here, if you look closely here, this is 1 triplet of microtubule. There are 3 microtubules here, and there are about 9. This is the second one, third one, 4, 5, 6, 7, 8, and 9, right? 9 of these. So 9 groups of these triplets. So there are 27 microtubules. So these microtubules join alongside each other in a cylindrical form to form the centrioles, right? This is a bit complex, but I hope I made it clear for you guys. This is how your centrioles are made. Do not think that centrioles play a role as MTOCs, deciding whether to break down or make up microtubules. That is wrong. That used to be believed by scientists before, but now it is discovered that it is the centriole that acts as the MTOCs to decide whether microtubules need to be made or broken down. Centrosome has no particular, function is just an area in which your centrioles are present, and the microtubules are basically radiating, or they basically come from the centrosome. So just understand that. If you look at the book from Mary Jones, that's what it said there as well, all as per the syllabus. Okay, so let's see if there's more. Okay, that's it for today. That is it for today, guys. I hope you enjoyed this video. I really hope that you guys understood, because we have successfully covered the organelles that are found in both an animal cell and a plant cell, and we have also successfully discussed and seen their structures, functions, diagrams, and everything else. Then the next video, I'm going to be making a video on these organelles found in a plant cell only. And I'm also going to be making a video on microscopy, in which I discuss about the eyepiece, graticule, some past paper question videos, and I will be continuing with O levels in IGCSE biology as well. I'm not yet done with that, so I'll be continuing with that as well. So guys, subscribe to my YouTube channel, like, share, follow and like my Facebook page, comment down below, react to my video, share this around, and if there is any help or any request, I'm always there. Comment down below if you need any help, right? Share this around with as many people as you can, because this would mean a lot to me. Your support means a lot to me, and we are growing. That is correct. We are growing, and it is all because of you. So thank you so much, guys, because we are growing, and I hope that we grow even more. And the more we grow, the more the better videos I'll make for you guys, and the free content and amazing helpful tips I will give you guys. So that's it for today. See you next week.